Not yet, right? Okay. So, okay, let's get going. So, hello everyone. So, welcome to another VacCAP webinar. Today, our guest is Felipe Ferrão. He's a research faculty at University of Florida with Dr. Patrizio Muñoz's team. Felipe is an expert on quantitative genetics and blueberry molecular breeding, and today is going to talk about the work that we have been doing in VacCAP to understand the genetic mechanism controlling volatiles in blueberry and the link to flavor. Before we start, just a couple reminder. You can post questions on the chat while Felipe is presenting, or you can wait the end of the webinar to unmute yourself and ask questions. And after the webinar, we will have a survey that we will ask if you can take it. With that, I guess we'll pass the ball to you, Felipe. You can take from here, okay? Thank
Hello,
you.
Massimo. Hello, everyone. Massimo, thank you for the invitation. My name is Felipe. I'm a research scientist in the blueberry breeding program. What I'm going to present here, it's a kind of collective effort to put together some slides combining like blueberry flavor and also quantitative genetics. It's part of this presentation. I have Patricio, Camila, and Juliana, and also, of course, like all the VacCAP team um, that help us to collect data and also some results. Okay, so the title of my presentation today is a multi-omic roadmap to understand blueberry flavor. Um, I would like to start this presentation with a short background about myself. So I'm a biologist by training, and then I joined the blueberry breeding program first as a postdoc in 2017, and then later I was hired as a research scientist and helping the blueberry breeding program to combine statistics and genetics to take decisions for breeding. So I have one simple slide just to tell you about like why blueberry is so important. Blueberry is the second most important soft fruit. The per capita consumption has increased more than 97% in the past 10 years. And there are two main reasons for that. Number one is because it's delicious, and number two is because of health benefits. So blueberry has a status of superfood, given the health benefits that are associated to the crop. Um, there are like a couple of challenges on releasing new cultivars. And the first point is because breeding is complex, and I have to mention this. So it's very time consuming. So on average, using traditional approach, it can take like 15 years to release a cultivar. It's a perennial species. There is a high level of inbreeding depression, and the species is also polyploid. So in this context, the University of Florida has a long history on releasing cultivars or releasing what is so-called like southern high bush cultivars. And just to give a number, so from almost 200 cultivars that they were released from 2002 to 2018, 79 they are coming from Florida. So um, the main PI of the 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 main breeder is Patricio Muñoz. Here, I want to point out something that is really interesting and something that is unique to the breeding program is because we have those two branches working like side by side. That is research and breeding. So everything that we have at the research level, we try to bring to breeding decisions. Um, so just to represent a little bit about like the importance of the cultivars or the UF cultivars across the globe. So here we have different breeding programs that they are like blueberry breeding programs, and as you can see that most of them they are using materials or they are using pedigree from the University of Florida. So just to give an idea that the University of Florida blueberry cultivars they are everywhere. Regarding research, the blueberry breeding program here at UF is mainly divided in four main sections, and it's interesting how the data or how the information flows among the groups. So we have one group that we call it phenomics, that is basically is going to design like robots and use computer vision to better um, to improve or leverage the data collection. So all this information goes to my team, that is the statistical genetic team, that we're going to use all this information. We're going to combine with omic data to run genomic selection, GWAS, and test like all these different kind of models. So everything that is interesting, we move to the computational biology team that is going to go further and take a look on potential candidates, potential genes, and if we define any important target, we move to the molecular biology team, that is this team here, that is going to run like the protocols for gene editing, CRISPRs, adapted tissue culture, and everything that we are doing here, again, is to support breeding. Okay, so this is more or less how is the structure of the blueberry breeding program at the University of Florida. So in the statistical genetic teams, we have been working with different traits, as you can see. And it's interesting that like all these theoretical outputs that we have for like understanding the genetic architecture of some specific traits or using simulations to support the breeding program, we try to translate it into practical outputs. So when we say like practical outputs, it could be softwares, it could be modules, it could be candidate genes. So the idea is have a better understanding about like key different traits that are important for the blueberry consumers, from the blueberry industry, and having a better idea uh, about these traits, we can move this to practical outputs or try to guide breeding decisions. So on this framework, among the different traits, among the different practical outputs that we have, flavor is an important trait for us. So I want to start this presentation, or I want to start introduce the concept of flavor into six main questions. So when I start to work with flavors in blueberries, we created or we designed those six main questions that we should be able to answer to actually have a better understanding about this trait. So number one is, what is flavor? If blueberry flavor matter to consumers, what are the components, what are the chemicals that sometimes created this aroma perception in blueberries? What is the genetic architecture of blueberry aroma? If we can predict consumer preference, and how we can use multi-omics, or how we can combine different sources of information to accelerate flavor-focused breeding. Okay, so let's start with the first question. That is the basic question. So what is flavor? So flavor in a very simple way is the sum of inputs from multiple senses that is going to inform to our brain what we are eating. So every time when we eat something, there are like two main kind of information that we're going to be able to understand. So number one is we have receptors in our mouth that is going to recognize what is sweet, sour, salty, bitter, and umami. So this is the first source of information that we call like taste. Okay, so there's another second important source of information that is the aroma. So the fruits or the foods, they gonna release some volatiles, and those volatiles is gonna leverage our perception about smell or about aroma. So the exercise that I'm gonna propose here is what happens when we eat something and we are cold. So you're gonna see that the perception of the flavor of the food is not the same. And the idea behind is because our nose is kind of blocked. So then we cannot realize about the aroma. Okay. So the message here is flavor is a combination between taste and aroma. So So this is the take-home message. The first one that I want to deliver in this talk, that flavor is complex, is a multi-dimensional experience that is going to come from the interaction of multiple different senses. Okay, so then comes to the second question: if blueberry flavor matters to consumers, and the best way to understand about this is we should ask them. So this is a paper that was published in 2014. And if you can look here, the abstract of the paper, so they run this kind of evaluations among consumers, and 61% of the population uh, that was asked, they said that flavor is the most important trait in blueberry. So the point is, if you go and talk with consumers and ask them, okay, so based on everything that you know about blueberries, like health components, the shape, colors, uh, flavor. So tell me the texture. Tell me what is the most important trait that we have in a blueberry, and base it on not only this study but multiple studies. Flavor is usually is the top number one. So consumers, they care about flavors. Okay, so blueberry flavor is a top priority among consumers. So with that, we start to enter more and more on this uh, um, on this concept of flavor in blueberries. And one of the first studies that we run formally addressing flavor at the University of Florida, it was back to 2017 with a PhD student. And what was really interesting is that for years and years, breeders, when they go to the field, they were testing some blueberries, and they were describing some blueberries as aromatic, as sweety, as fruity. So we start to see that there are like some new kind of flavors that we could describe blueberries. So what happened is, um, so based on this kind of information, we selected like 60 genotypes, and then we divided them in two main groups. So we have genotype that consistently breeders are calling as aromatic because of the sweet and floral perception, and genotype that we always call it as non-aromatic. And this aroma classification was done like by breeders and also by sensory panels, and we decided to collect the volatiles and the chemicals. So this paper was published a couple years ago. So, and there are a couple of results that are interesting that start to show us like what is flavor and what is the diversity of flavor that we have in blueberries. So the first point is when we divide these two group of genotypes, so genotypes that they are classified as aromatic and a group of genotypes that are classified as non-aromatic, this is we submitted, we send those group of genotypes for formal sensory analysis. So essentially for 2 different years, we set up like sensor analysis in the blueberry at the University of Florida, and we invited more than 500 consumers, and they taste. And then they start to give scores for flavor liking, juiciness intensity, overall liking, sourness intensity, and sweetness intensity. And what's interesting is that all the genotypes, I mean, statistically, the genotypes that the breeders classify as aromatic, they had a higher overall liking or than the non-aromatic genotypes. And here you can see more or less the kind of words that the people use it to, or the regular consumers, they use it to classify genotypes as aromatic. So when they were eating blueberries, they were classified as aromatic. Most of them use like words as fruity, floral, strong blueberry flavor. So this is something interesting. Um, and then the following question is, okay, so what is creating this? Why we have some genotypes that they have those kind of unique flavor profiles? So essentially, what we can see in this plot here, in this first plot at the left hand is when we put together all the sensory, all the the chemicals. When I say chemicals, I'm saying like sugar and acids and volatiles. We can see that the two different groups, the aromatic and the non-aromatic, they are in different portions of the PCA. So when we move to this direction, so we can see that a group of terpenes, they are associated to the aroma perception. So that was the first indication that technically what is creating this kind of aroma perception that consumers and breeders can perceive is created by a small group of terpenes that is enhancing the ability of consumers and humans to recognize or to feel that blueberries, they have those kind of specific notes that we call it aromatic, but you should think about this as like floral, uh, fruity notes. Okay, so this is the take home message number 3, that yes, it seems that we have a group of terpene volatiles that can enhance our aromatic perception. Um, the following question that we had is, okay, so now that we know what is flavor, we know that flavor matters, we know that we have some specific flavor nodes. So what is the genetic architecture of the blueberry aroma? So to make it, we have a large breeding population with 1,000 of blueberries. We genotype everything. And at that point, we phenotype it only for 15 volatiles. Uh, and then we run like GWAS and also genomic prediction models. And the paper was published 5 years ago uh, um, in this journal. Okay. So the first point is when we run the GWAS for all the 15 volatiles, we saw like for every volatile, for all the volatiles that we tested, we saw this kind of tower-like structure. So it means that so this is one example. This is the volatile that is called 2-andecanon. A single marker here at the top by itself is explaining more than 30% of the phenotypic variation on this volatile. And we saw this for most of the volatiles. So the underlying hypothesis is that volatiles are traits with simple genetic architecture. So it seems that they are controlled by few genes or focal genes across the blueberry genome. So with that, we start to make some tests, or we start to study like how we can approach this at the breeding level. So what I'm showing here is a PCA, and we have two different colors. So the color in gray and the color in black. The color in gray is we have around of 900 genotypes that I call it population 1. So in this population 1, that I call it, we run all the GWAS analysis. We identify like all the hits. Later, we included another population that we call it as population number 2, that is presented. Each individual is represented as a black circle um, in this PCA, and we use those kind of individuals for validation. So we never mixed the population 1 and 2. Okay, population 1 was for discover, G was inference, and population number 2 was for validations. Okay, and then we start to run genomic prediction models. So essentially, using only the population 1, that it was the original population, we run a cross validation and we start to predict all the volatiles. So, and what we can see is that the prediction ability using traditional genomic selection approach, in this case, we use like RR-BLUP, we can see high prediction ability. That is something that is we expect when we have like traits that they have a simple genetic architecture. Just to give you a number, so if you try to run genomic selection the same way, but for traits like yield in blueberry, this value is going to be close to 0.2, 0.15. Okay. So the second exercise is, okay, what happens if I train my model in the population 1, and then I predict my population number 2. And as we can see in yellow, we have a reduction in the prediction ability. That is something that we already know that has been proved for many traits in different crops. So the next scenario was, so what happened if I use the GWAS information or the hits that we could find in the population 1 as fixed effect or as prior biological information to predict the population number 2. And this is a true validation. This is a true cross-validation. Because why? Because we never use like the population 2 for the GWAS to avoid any kind of overfeeding. And as we can see, for most of the traits, when we compare like green and yellow, we increase the prediction ability. So it means that every time or most of the time that we bring or that we have like prior biological information and we include those kind of information in genomic selection, the prediction ability is going to increase. So the very last scenario is we move from the GWAS directly to prediction. So instead of running like genomic selection models, so we just grab the 5 largest hits that we identify in the GWAS that we run the population 1 to predict the volatiles in the population number 2, and we have very nice results. So if you come here, you're gonna see that linalool. Linalool is a volatile that is very important in blueberry that enhance our floral perception. So if I try to predict the linalool using like only 5 markers, I have a prediction ability that is like 0.36. So it's another good evidence that actually the aroma or the volatile in this specific case has a simple genetic architecture. So um, with that, so the take-home message from this work is the volatile has a simple genetic architecture. And it means that in terms of breeding, this is a very good news. So the next question is if we can predict consumer flavor preference using metabolomics. So far, we are using genomics to have a better understanding about the metabolites or try to identify what are the genes or markers or candidate regions that are controlling the metabolites. But now the question is, okay, so I want to use this to predict how much the people are enjoying blueberries. Um, and this paper was published in PNAS 3 years ago. And the exercise is kind of simple. So if I come to you and say like how to evaluate flavor, so your probably your answer is going to be okay. Just uh, um, just provide consumers with a sample and then ask them. So this is the concept behind of the sensory panels. So we have like real consumers. They are tasting like blueberries, tomatoes. They are testing different foods, and then they start to put like scores for overall liking, sweetness, sourness, firmness, flavor intensity. This is the simplest and a precise way to measure this, but there are limitations. The first limitation is because it's expensive. So just to give a number, this is probably the most expensive phenotype that we have been collecting in the blueberry breeding program. So in each sensory panel, we can include a maximum of 5 genotypes. And there is a reason for that. If you present to the consumers more than 5 and they start to eat and test, at the end, they're going to taste like all the same. And then we're going to mess up the evaluation. Okay. Um, I'm sorry. Um, the second point is because it's time consuming. So in each sensory panel that costs close to 5,000, and we can include like only 5 genotypes, it means that uh, each phenotype is going to cost 1,000 for a breeding program. So it's expensive and it's time consuming, organizing like all those sensory panels, and it's low throughput. So the idea behind is what happened that at the same time that we move the samples to the sensory panels, we move part of the samples also to the lab, and then we collect information for chemical, and then we have like information about the sugar, the acidity, and also the volatiles and the texture. And then how can we predict, or the question is, how good is going to be my prediction, or can I predict how much a consumer is going to enjoy a berry if I know like what are the chemicals and genetic information? And this is the exercise that we are doing right now. So what we did was we organized a historical data set with tomato and blueberry, and we tested a different machine learn and statistical approach to predict the sensory preference. Okay, so we had like close to 200 samples in tomato, um, about like 250 and in blueberries. We run these in multiple sensory panels, so we have historical data in the case of tomato from 2010 to 2015, in case of blueberries from 2012 to 2017. So here you can see the number of panelists that I mean the number of consumers that show up to test like those different samples. And the exercise here was, okay, so the consumers, they are scoring the tomatoes and the blueberries in terms of overall liking, sourness intensity, sweetness intensity, and flavor intensity. And the question is, what is the importance of the sugar and acids, and what is the importance of the volatiles? And as you can see here, for blueberries, and we're going to focus on this plot here, more than 50% of the variation in terms of overall liking was explained by the volatiles. It means that the volatiles, they have more importance in blueberries than sugar and acids to tell about like the overall liking or the sensory perception from the consumers. Okay, so we know that volatile is very important, but the question is how much I can predict of the consumer preference. So in this plot here, we have different statistical methods that I presented on the rows. And in the columns here, we have basically the two crops, so blueberry and tomato. So, and here we have the different evaluations from consumers. Okay. So if we look this value here, there's telling like 0.7. What I'm telling you is that using this statistical or this machine learning method that is combining like all the volatiles and all the chemicals in the same model, we can predict with 70% of accuracy if a consumer is going to enjoy or not a blueberry. So the point here is tell me what are the chemicals, or tell me what are the volatiles and the sugar and acid of a given berry that I can tell you with around of 70% of accuracy if a person is going to enjoy or not, or if the consumers on average, they're going to enjoy or not that blueberry. Okay. Uh, we tried to do the same exercise, but at that time we had like data only for tomato, and we compared genomic and metabolomic selection. So can I predict, for example, overall liking? Can I predict better using the metabolites, or can I use or using genomics? And what we can see is that for all the traits, if you have information about the metabolites, we're gonna have higher prediction ability. Okay, so that's the take home message number 5, that consumers liking or the sensory perception can be predicted using metabolomic information. So then we come to the last question, that is like how can multi-omic approach accelerated flavor breeding? And we have all the work that we have been doing with the VacCAP. And in this specific section, we have we put together a great team with USDA, University of Florida, and NC State. And the central idea is, okay, based on everything that we did, everything that we already know, so how we can expand this using like large number of samples, and also using diverse group of blueberries. So this is what I call it VacCAP flavor. And essentially, before like entry or before like jumping to the results, I really would like to show this framework because this is how the information flows in a biological system. We have the genes or the genomics that they're gonna be able to codify the metabolites. That in this case, those are the volatiles and also the sugar and acids, and those kind of things or those kind of chemicals, they're gonna impact the sensory perception. So we have genomics and we have metabolomics and we have GWAS and genomic selection um, in multiple layers. So we can use genomics to predict directly the sensory. It's very difficult, very hard. Uh, we presented in the last minutes how we can use metabolites to predict sensory, that we call it metabolomic selection. Um, we also have genomic selections for volatiles and GWAS for volatiles. That was also part of the results that I also presented in this seminar. So I just want to show you that move from this part or move to the genes directly to the sensory. It's something that is very complex. So this is the reason why we try to have a better understanding about like the chemicals and the genetic architecture behind of the chemicals that is gonna affect the at the end of the day the sensory. Regarding the VacCAP, now we have two different populations. So we have northern high bush blueberries and we have southern high bush blueberries in two different breeding programs. Um, one in Oregon and another one in Florida. And we have genomic information. So we use the LGC genotyping platform. We call like all the allele dosage, and as you can see here, the two different the northern high bush and the southern high bush, they have very different like genetic profiles. We can see a strong structure, or you can see that they are very different genetically speaking. At that time, one of the is my main expertise is not about biochemistry, but at that time we had a kind of big challenge because we want to run volatiles for more than 2,000 genotypes. Um, and we spent like more than 2 years working with different institutions and different universities. And then we set up this collaboration with the University of Alberta, Canada, to adapt blueberry protocols for high throughput data collection. Um, and then we decided, so at that time we tested like dynamic headspace versus SPIMI. We test like different kind of approach that we could actually extract volatiles in a high throughput way. So this was one of the paper that we released, but it was good. But because prior to the VacCAP project, we could define a good protocol for high throughput um, um, volatile extraction. So this is part of the results. So in total, we have information from 68 volatiles, and this was really interesting because here I put in a kind of proportion. So all the values is going to range between 0 and 100, just to give you an idea about the proportion of the volatiles across the northern high bush and southern high bush. So in total, we could find like 20 volatiles that they were like uniquely represented in southern high bush in the population that we have in Florida. So they are presented in red, and as you can see, they have like those kind of volatiles. They are presented only on the samples or on the genotypes that we have in Florida. We have just one unique volatile that we could find only on samples in northern high bush. So that was the first nice results, like how northern high bush and southern high bush, they have like different profiles. Uh, Also interesting, we start to look the volatiles across like both populations, southern high-bush and northern high-bush, in terms of pathways. Um, and we can see that the volatile pathways, they are more diverse and well distributed in southern high-bush. That is this part here. So, and when we look like northern high bush, they are rich on oxalipine and the branch chain volatiles. So it's interesting to see, like, again, how the two different populations, the two different samples, one from Florida, another one from Oregon, southern high bush, northern high bush, they have like different profiles. So the next step is, okay, so we have this number of volatiles. They are coming from different pathways. So let's run GWAS. Let's see a little bit more. Let's try to get some insights about the genetic architecture. So in this plot here, you can see the different volatiles that they are presented with those small circles, small bubbles in front of. They are coming from different pathways. They are presented, each pathway is represented by different colors. But the attention here, it should come to the bubbles. So we have 12 bubbles. So if you come and look here, for example, we are looking, for example, the hexanol that is coming from the oxalipine pathway. And then we have three different colors. If the color is blue, it's because we could identify a gene or a region or a marker that is significantly associated to the trait. Only for, if it's blue is only for southern high-bush, if it's green is only for northern high-bush, if it's in red, it's because it's in both cases. So in this specific case here, we have in the chromosome 10, that is the bubble number 10 that I'm presented at this point here, we could find a significant genomic region that is associated to the hexanol that was presented in southern high-bush and northern high-bush. Okay, so I want to put some attention on the eucalypto example, and there is a reason for that. So this is a simple correlation plot. On at this point here, the plot, we have the sensory traits, like when consumers, they are eating blueberries, they are scoring for liking, sweetness, texture, intensity, and sourness. And then we have here different chemicals and volatiles. So what is really important is, as we might know, is that sugar, when the blueberries, they have like higher amount of bricks, most of the consumers, they're gonna enjoy the berry, and so there is a positive correlation with liking. And acidity that is presented here as a TA has a negative impact. So in general, consumers, they don't like blueberries that they have a high acidity. So, and we can see this when we compare like two different cultivars that we have in the breeding program. So we have Kestrel, that you can see that the liking that is presented in this part here of the spider plot is really high, and Windsor, that the liking is pretty low. And what is creating this is technically the amount of acid and the amount of sugar. So we can see that Windsor, when we compare to Kestrel, it has a high acidity. So the acidity is basically is controlling the overall liking. But take a look at this. We have a second example that the amount of sugar and the amount of the TA is very similar. And I can tell you the texture is also similar. Uh, but people, they really like Kestrel, but they don't like too much is no chaser. And there is a reason for that. One of the reasons is because no chaser is one of the cultivars that we have in the breeding program that has the highest amount of eucalyptol. And we have been seeing this for years and years. So all the samples, most of the case, that they have like high amount of eucalyptol, people, they don't like. So we have one example of a volatile that has a very simple genetic architecture that when it's present or when they are in high concentration, the people, they don't like, even if the amount of sugar and acids, they are in a good balance. Okay. So, and this is what is interesting about eucalyptol, and this is why I put like the eucalyptol examples. So this is the historical data that we have in the breeding program in the new phytology paper that we published like 5 years ago. So for the eucalypto, we have basically two hits, one in the chromosome 2 and another one in the chromosome 11. And all these results here that I'm presenting here are coming from the VacCAP. So we have a different population, southern high bush, a different population, northern high bush, and then when we combine like both informations, and we can see exactly the same hits. So this is a kind of clear candidate of a volatile that is impacting consumer preference, and they have a simple genetic architecture. Okay, so why I'm presenting like all these results and how we can start like to combine informations and how we can start to use all those things to help breeders. The last result that I want to present about part of the VacCAP, it's kind of connected to one of the papers that we published in 2022 that we show how we envision that flavor studies they should be addressed in the future. So we believe that breeding programs, and this is what is happening right now in the US breeding program, and also as part of the VacCAP, that we're going to collect data in multiple layers. So we're going to have genomic information, we're going to have metabolites, we're going to have transcriptomics, we have environmics data. In the case of the VacCAP, we're going to have texture data. So we have information from multiple layers that we call as multi-omics. And the best way to get to the final target, that is to predict consumer preference, is like combining all those multiple layers. And this is the exercise that we did. So when we start to run when we start to run or when the VacCAP trials at the University of Florida, we collect not only the volatiles, but we also the genomic information, the chemical information. So we have information about the breaks, the TTA and the PA. So we know about like the sugar and acids. And also we start to collect the texture data. So we use the texture analyzer to have a better idea about the texture of each one of the genotypes. We run this for more than 1,000 samples, and Julian and I, like two researchers at the University of Florida, we try like berry by berry, and we score them for liking, aroma, and texture. So at the end of the day, um, we use like all this information to try to predict the flavor preference. And the results are kind of interesting. So here we have the three sensory traits that we have that we score technically, the flavor liking, aroma intensity, and the texture liking. And this is what happened with the prediction ability um, when we start to accumulate different kind of layers. So for example, if we use like only genomics, we can predict the overall liking or the flavor liking with less than 40% of accuracy. But this will happen when we start to accumulate all the results. So, okay, so what happens if I have like the genomics and the chemical? If I have the genomics and the texture, and if I have the genomics and the volatiles, and when we come to the end, so what happens if we start to accumulate in a single approach or in a single framework information from genomics, the chemical, the texture, and the volatiles. And we can see that the prediction ability, every time when we start to put like more and more layers of information, the prediction ability is start to increase. So this is what I like to say, like how information is powerful. So what is the importance of combining multiple sources of information, because each piece of information is going to explain a little about like a kind of complex target or kind of complex trait. Okay, so those are the main take-home message. This is a work in progress. We are still like analyzing the data to publish the paper, but so far those are the take-home message from the VacCAP project. So number one is we have like different aroma profiles when we compare southern high bush and northern high bush. Um, this confirmed like most of the studies that we did a couple years ago that the volatiles, they have a simple genetic architecture. We saw for some of the traits, we could see like consistent hits across different population studies. And this is the example of the eucalypto that I just presented. And those markers, we are called as candidate region because we are already designed like CASP markers for marker selection. Okay, so a year ago, we designed like CASP markers for eucalypto, and we genotype 20,000 of seedlings in the greenhouse for high eucalypto and low eucalypto, and those kind of information is helping to guide the breeders. Okay, and again, um, all the every time that we combine like multi-omic or multi-layer or different kind of layers of information, we saw a trend to increase the prediction ability. So um, this is the one of the my last slides. So in terms of conclusion, I would like to present what I call it roadmap. Okay, so if I could summarize uh, part of the VacCAP research in terms of flavor, this is the roadmap that I wanna present as one of my last message. So everything started here when we have like better molecular tools. So it's part of the VacCAP, assembling like different reference genomes and also a pangenome. So some of these results have been published, and then we designed a genotyping platform. So from here, we have two different directions. So this path here is what I call it the path that is the non-traditional path that we are measuring like new traits as part of the VacCAP. So we have texture that has been analyzed by texture analyzer, and we have also the volatiles. And here, at this point here, we have the conventional traits like sugar, acids, and firmness. So at the end of the day, we are combining those two sources of information um, into a single path here. So in yellow, we have the sensory analysis. So we have been running or we have been collecting sensory data as part of the VacCAP in different directions. So we have regular consumers, so we have information from breeders and also training panelists. So when we put like all together, those kind of omics information with the sensory, the ultimate goal is to deliver like breeding tools. They're like gene discoverers, it could be models, softwares. Um, so try to design tools that breeders can use to guide their decision. Okay. And this decision have been made in two different directions. So when we have like some traits that they have a simple genetic architecture, there's the case of eucalypto. There are some papers showing that the acidity is also controlled by a single region. So this could be used for marker selection. So for traits that they are a little bit more complex, we have been using either genomics or metabolomic selection. So here, what I call it is future research is like, okay, point some of these key regions, and we can move this to gene editing that is still not part of the VacCAP, but is part of future research. And the idea is, at the end of the day, use like all these tools or walk through those different paths to have a cultivar. A cultivar that is better for consumers, that is better for growers, that is better for stakeholders. Okay? So once again, the idea is combine all this information, and here I'm talking like more about flavor to deliver tools that we can use to release cultivars. Um, this is my personal point about like all these studies. So the question is what I have learned in the last few years of working with flavor. So number one, this flavor is very complex. It's a trade that is very, very complex, especially because we need like people to score flavors. And you know that people, they have different backgrounds, they have different opinions. Sometimes they have they cannot express themselves. So it's very complex. Um, I have so I want to point out or I want to highlight the importance of using multi layers of information. Um, and one point that's really interesting is how like breeders, geneticists, people from food science, statisticians, they have a lot of to contribute in terms of experimental design, in terms of methods and softwares for prediction inference, in terms of functional validation. So creating a kind of multidisciplinary team as we have in the VacCAP, I would say that it is one of the best way to keep moving this kind of research line forward. What is the future? The future for me is crops, they have different demands and objectives and tools of evaluation. And we are dealing with a new willing to experience and pay for new flavors, aroma, texture, shapes, and color. It makes like flavor a very important trait. Okay. So I want to take this opportunity to say thank you to all the Blueberry Lab, to the Blueberry team. So inside of the Blueberry team, there is a specific group of students, research scholars, postdocs. They have been working directly with me. Um, this is also part of most of these results is part of a great collaboration. There was part of the VacCAP, but also data analysis, interpretation, discussions with colleagues from USDA, Florida, NC State. Okay. So with that, I want to thank you, and I'm glad to take any question. Thank you.
Thank you, Felipe. Great talk and great overview of what has been done and what we think that we can do next. Let's open for questions. So everybody, you can either unmute yourself and you can just ask a question, or you can post a question on the chat.
Massimo, this is Jim. Can I ask the first question?
Yes, go ahead.
So Felipe, I'm curious if you have a feeling, and I'm sorry if I missed it, on how much of an environmental impact there is on the volatiles in particular in blueberries.
Jim, thank you. I want to say that volatile, they are very volatile in the sense that the environment plays is very important. So far, it's on part of the VacCAP. We have some work in the blueberry breeding program evaluating volatile across the post harvest. And so we have volatiles measure across different time points, like when we harvest the berries after 1 day, after 1 week, 3 weeks, 5 weeks, and 7 weeks. And in general, so in this specific condition that is post-harvest, there is a reduction in the volatile expression, and the interaction is very important. I think the biggest challenge here is the volatile is still expensive to be collected, is still hard to collect, and set up those kind of multi-trial or multi-environment trials for volatiles created one more layer of complexity that is the evaluation of the trait by itself. But my answer is G by E, it's definitely very, very important.
Thank you.
Good question.
I have another one if everybody else is still thinking.
Go ahead, Jim.
I was curious about which northern highbush population was used, because a lot of northern highbush now have a certain amount of Vaccinium darrowii in them, even a little bit of Vaccinium angustifolium. There's almost no modern highbush cultivars that are 100% hornbosum.
Mm-hmm.
And that, I know that adds another a layer of complexity to all your analyses. I'm just curious if you looked at some that were known to be more more interspecific hybrids. If you'd find not necessarily different volatiles, but of course, I'm thinking some of the northern highbush might be now closer to some of the southern highbush, now that there's introgressed Deroi into a lot of the northern high bush cultivars?
There's a very good question, and actually something that I need to pay more attention. So we have the genomic profiles of both populations, and we can try to answer this question. So um, I have more familiarity with the southern high bush part compared to the northern high bush. But what you're asking makes total sense, and we can approach this by looking the genomic profiles and also the when we run the structures and the PCA. So um, we can definitely we can look for this and also check how the volatile profiles are changing.
All right, I have one more question.
Sure.
Go ahead.
All the blueberries we're dealing with are either cultivars or came out of a breeding program. And so in theory, they've all been selected to some degree, right?
Yes.
So what do the natives look like? What is a native? I noticed I guess there's no such thing as a native southern high bush, right? Because they're all interspecific hybrids. I just wonder what how some of the more wild material looks as compared to some of the more the cultivars or selected material, and I wonder if it can start to give you an idea of volatile preferences, because in theory there has been selection for those flavor components over time, right?
That's a
So if
very
you look at unselected material, it might give you a clue as what's been selected for.
that that question is kind of complex in two different sense. Number one is, and I'm gonna use the example from tomato, and then you can correct me if you disagree. But what happened with tomato is that most of the selections they were never they were driven by production and not by flavor. So even if they have like tomatoes that they taste like pretty well, but they have a kind of poor yield performance, uh, they had issues on the selection process. So what I'm trying to say is like, at least in the past, um, flavor was not a top priority, and I'm using tomato as an example to illustrate this. So this is the first part of my answer. Okay. So the second part of my answer is um, what you are asking is like in the blueberry breeding program, the first criteria. So I'm talking about Florida. The first criteria was always like flavor. So why? Because we have like visual selections, we have breeders going to the field, they are like tasting the blueberries, and if something is really bad, they were never selected. So, and what I can tell you is that like going to the field and working on those first generations of breeding, we have like flavors that they are awful. So we have some cultivars, or we have some, not cultivars, we have some samples that we call like as wet dog, because the flavor it's really bad. So to answer your question about like how we can connect the wild flavors with the flavors that we have on the new cultivars, what we are doing right now is we are running also volatiles for wild species. So there is a student in the blueberry breeding program right now, and she's working with wild species. She has been collecting samples, and the plan is start to uh, also including in the to see the volatile profile. So I think with those two pieces of information, I would say three pieces of information like northern high bush, southern high bush, and the wild species. And then when we start to see the volatile profile, I think that we're going to be able to start to connect the dots.
Yep, sounds good. Thank you.
Felipe, Juliana here. Can I just like complement your answers?
Sure.
Go ahead, Juliana. Go ahead.
Yeah, I think like in the blueberry breeding program, we have more diversity of volatiles when you compare to the northern population, right, Felipe?
Yes.
Yes, I think maybe this diversity, this increased diversity, the population of northern hybrids from Oregon that NALA has provided. And then in southern high bush, Vaccinia elliottii was one of the sources of aromatic compounds that has been used, and we have some cultivars with some Vaccinia elliottii in the background. They are very aromatic.
That's a very good point. Very good point. I know that sometimes for people that are listening, this is the complexity of flavor, right? So they have questions about like what is aromatic, so what those people are talking about. So there are some blueberries that technically when you eat and consumers, regular consumers can perceive this, it was part of the research, they kind of enhance or our ability to recognize what is floral. So it comes to my mind or to the mind of the consumers like floral fruity notes. So this is what we are talking about when we say aromatic.
Okay. I guess is now this framework helping the GWAS helping to start to pinpoint that some candidate genes for some of the compounds, Felipe?
Yes. So one of the points is like as part of a discussion, so you can see like how the subjective, how complex, how expensive is to measure those traits. So imagine if we have a marker that can predict or can explain um, um, one of the volatiles or one of the aromas that plays an important role in consumer preference. So the GWAS, so far, we have very solid evidence about the eucalypto. That is the example that I presented. So it means that we use like all the GWAS to map, narrow down different regions for eucalypto. Then we design different markers, tested in a population. So it's working. So what you are doing right now is using this information for marker selection.
Thank you. Okay. Yes, just a last call if there is any question. You will receive also a survey. If you can please take the survey. And yeah, that's the note from Michael. Make sure that you answer the survey. You should receive in your email address. And just another reminder while we wait, look up for the upcoming announcement for the July webinar. Hector Lopez Moreno will present from Yuan Zalaba Lab will present about cranberry texture genetics. There is no other questions. Thank you so much for attending the webinar. Thank you, Felipe, for your talk. Bye-bye.
Thank you. Have a good day. Thank you.